Translation is the process by which the genetic information carried by mRNA is decoded to synthesize a protein. It occurs in the ribosomes, the cellular structures responsible for protein synthesis. Here's a detailed overview of translation. Initiation: the process begins with the binding of the mRNA to a ribosome. The small ribosomal subunit attaches to the mRNA's start codon, August, which codes for the amino acid methionine, or another specific amino acid in certain cases. Initiator tRNA, carrying the appropriate amino acid, binds to this start codon. Elongation: during this phase, the ribosome moves along the mRNA strand, reading the codons one by one. Each incoming codon is recognized by a complementary anticodon on a specific tRNA molecule. The ribosome facilitates the formation of a peptide bond between the amino acids carried by the adjacent tRNA molecules. The ribosome then shifts along the mRNA, moving the tRNAs through the A aminoacyl, P peptidyl, and E exit sites. Termination: the process continues until a stop codon, WA, WAG, or UGA, is encountered on the mRNA. These codons do not code for any amino acids but signal the end of protein synthesis. Release factors bind to the ribosome, leading to the detachment of the newly synthesized protein from the final tRNA molecule and the dissociation of the ribosomal subunits. Post-translation modification: once the protein is synthesized, it may undergo various modifications to become functional. These modifications can include folding, addition of chemical groups, such as phosphorylation or glycosylation, and sometimes cleavage of specific segments. The result of translation is a newly synthesized protein that has a specific sequence of amino acids dictated by the sequence of codons in the mRNA. This protein can then carry out various cellular functions based on its structure and function. Keep in mind that the process of translation can be quite complex, involving numerous factors and regulatory mechanisms. The details can vary depending on the specific organism and cellular context.